you need to support the emergence of life. Yeah, the second ingredient is an original life form. So in other words, an initial condition. And the third ingredient is simply like an algorithm, in this case, Darwinian evolution. So with these ingredients in mind, we can revisit the question and ask a more precise one. How can we create an artificial system, an initial condition, and an algorithm that can bootstrap itself to produce unbounded diversity and novelty? So first, we'll have a look at artificial systems. A cellular automata is a discrete model of computation, actually one of the simplest models of computation. And despite their simplicity, they have become fundamental in understanding and studying complexity and emergence. A cellular automata consists of a grid of cells. So you have a state, here with discrete values between 0 and 1. And you also have a rule. So basically, the rule is fixed, simple, deterministic, and you basically just take your state, you apply the fixed rule, you get a new state, and you do that over and over again. A prominent example of cellular automaton is Conway's Game of Life. It was invented by John Conway in 1970. And it uses a very simple local rule. And the same way that scientists were amazed at the emergence of life from inanimate matter, mathematicians have been amazed at the array of diverse self-organizing patterns that were discovered within this cellular automaton. So here's an example of Conway's Game of Life, starting from a randomly initialized grid. And here you can see it in action. So you can see that the behavior is very lifelike, very organic. And soon after its creation, we discovered this pattern called the glider. And what is interesting with this pattern is that you can see that it moves. There is locomotion that is emergent property of the game of life. But there is more. Because with the help from computer simulation, large moving self-organizing patterns were discovered, just like this one. Or even larger, like this one. But why stop to such simple computational models? For example, the game of life is binary. And a cell is either dead or alive, which makes it difficult to reflect the true analog, continuous, and dynamic nature of the real world. So can we create a more expressive model that reflects more closely the real world? And the answer is yes. Lenia is a family of continuous cellular automata, and it generalizes the game of life. The algorithm is very simple. First, you initialize the world, and then you repeat the update given by this formula. So you can see that in the update, there is the growth mapping function G and the kernel K. And if you take those specific values for K and G, you get back like the game of life. But Lenia is much broader than that. And you can take neighborhoods that are much larger with larger kernels and also with smooth growth mapping functions. And indeed, if you do that, Lenia bridges the gap between the discrete nature of Conway's Game of Life and the continuous dynamics characteristic of the real world. So as an artificial system, we can see that Lenia enables the emergence of a diversity of lifelike, self-organizing, and robust patterns. Manual search led to the identification of hundreds of artificial species, uncovering emergent behaviors, such as like locomotion, reproduction, emission, and so on. So here are some examples of self-organizing patterns that were discovered manually by its creator, Bert Chan. So yeah, Lenia supports the emergence of diverse artificial life. And I think that it stands as a fertile ground for underlying the mechanisms of evolution within a controlled computational environment. So I think that as an artificial system, Lenia can serve as an ideal testbed for examining the emergence of diverse artificial life. So given that some high-dimensional complex systems such as Lenia support diversity of artificial life, we can revisit our initial question. And the challenge becomes how to automate the discovery of diverse self-organizing patterns within high-dimensional complex systems. To do that, we basically formalize the discovery of diverse artificial life as an evolutionary algorithm, and specifically a quality diversity algorithm. So now I will explain what is a quality diversity algorithm. If you think about it, nature encompasses numerous niches, and different species can be successful independently of each other in different niches. For example, the agility of wolves in hunting does not prevent whales to thrive underwater. So basically, in nature, there is no global competition, but rather a local competition that will foster diversity and novelty. Inspired by this idea, quality diversity is a family of evolutionary algorithms generating diverse population of high fitness individuals. So as usual, the algorithm requires a fitness function f, but also a descriptor function that will map the solutions into a lower dimensional space that will enable to compute distances in a meaningful way between solutions. So it will formalize the definition of niches. So in the end, quality diversity are simply genetic algorithms, but instead of doing global competition, you perform local competition, where the locality is defined based on the descriptors and fosters the niches and then diversity and novelty. So in this work, we will restrict the search space to world configurations that are two-dimensional of size 128 by 128 with 3 channels for RGB. And to give you an idea, here is an example of a self-organizing pattern in action. So that's the kind of patterns that we're looking for. We define the genotype as a seed and rule parameters. The seed is the center of the world of size 32 by 32. And the rule parameters are simply mu, sigma, and h, defining the kernel and growth mapping function that we were talking about. So to express the genes, it's very simple. You take the world, you take your genotype, you decode the seed, you put it at the center to initialize it, and then you take the rule in the remaining part of the genotype, and you use this to run the simulation. So each genotype will give rise to a different individual with potentially a different phenotype. Now, for the evaluation of those individuals, we will take the genotype, we'll express the genes. So we run the simulation, basically. And the first few steps will be considered to be the developmental process of the individual. So basically, those are discarded. For the remaining steps, we consider that the individual has converged into a stable phenotype, and we'll basically take measures on this phenotype to characterize the phenotype and define a notion of diversity. So for example, here I give a few examples like mass, color, angle, speed, and so on. And here I give also a few examples, an example of a fitness function and a descriptor function. So here the fitness function is defined as the negative variance of the angle. So basically we want to optimize patterns to go in a straight line. And the descriptor is simply the mass and the speed of the phenotype. So now we have everything we need to apply our first quality diversity algorithm. And we'll apply Maplitz, which is the most simple and most famous one, but yet it's very effective. So here we have our fitness function, our descriptor space with mass, speed. And Maplitz will start by discretizing the descriptor space into a grid of cells. And then Maplitz follows repeats the following steps. So first, it randomly selects an individual that you can see on the right, and it initializes the population with it. So you compute fitness, mass, speed, and you put it where it needs to go in the descriptor space. And then you randomly select an individual from the population, you take its genotype, you mutate it, you measure everything you need to know about this phenotype, and you add it in the population. So if you repeat that, you will at some point cover the descriptor space. And so now I can show you the results, the experimental results for this exact experiment. And if I show you the population at the end, we see interesting things. First, the descriptor space is fairly well covered. And you can see that at the bottom, you have the slow individuals. At the top, you have the fast ones. And from left to right, it's the so skinny ones to massive ones. So you can see that Maplitz here is very effective at discovering a diverse population of individuals with the specific characteristic of interest. We can do, of course, the same with different combination of fitness and descriptors. Here you can see that we optimize for the mass variance. So we want individuals that will grow and decrease in size. And here the descriptor is 3-dimensional. So here is just a sample. It's not actually a grid that we are looking at. And it's the color, so RGB basically. And again, we can see that Maplitz is very effective at discovering diversity of patterns with different colors, all optimizing for the mass variance. So one last example for Maplitz. Here we optimize for speed. And again, the descriptor is the color. And you can see once again, Maplet is very effective. Yeah, so basically Maplet uses manually defined diversity metrics to get the exploration towards specific characteristics, such as locomotion, mass, color, and so on. However, this manual definition, while it's good and interesting, it's also restricting the breadth of discoverable pattern. So how do we really break through and get a diversity of patterns, diversity of life? And the answer is that we can use unsupervised learning. So if you remember how Maplet works, first we define fitness and descriptors, and then we apply the loop of selection, mutation, addition. So now what we can do is, instead of manually defining those features, so those fitness and descriptor, what we can do is instead use unsupervised learning to learn that. We'll follow basically the same loop, but there is one extra step, that is the update of the parameters theta. And theta will then parameterize the fitness function and descriptor function. So the question is now, how do you update theta and how do you learn these unsupervised fitness and descriptor functions? So what we'll do is we'll use a technique from machine learning called variational autoencoder that is enabled to compress an image, so the image of a phenotype, into a low-dimensional vector. So think about the vector as a descriptor of the image here in green. And it does not store pixel values, but rather like a description of the image at a high level of semantic meaning. So to train this machine learning model, basically, it's very simple. Just take the image, you encode it, then you decode it with the decoder in red, and you basically minimize the loss between the input and the output. And then when you have this compression model, the encoder, you can use it at each time step. And it gives you like a latent trajectory. So at each step, you have basically a vector that is the latent representation of the image. And this will give you at each time step a trajectory of point in the latent space Z. And what is natural to do is now to take the average as the descriptor, because it describes your image. And as the fitness, you can use the negative variance of the latent trajectory, because the idea behind that is that you want patterns that stay coherent through time. So you want minimal variation within the latent representation of the individual. So here is a representation where you can see the green point is basically the average of the latent trajectory, and the red segments represent the variance of the latent trajectory. So we'll define the fitness and the descriptor. Of course, we don't need to use both these unsupervised functions. You can have a combination. For example, in this experiment, here we optimize for negative mass. So we want tiny individuals, and we use unsupervised descriptor. And you can see that we really have a much broader diversity, encompassing a wide array of shapes, colors, sizes, and locomotion properties. So it's significantly better than Maplet's. Here you optimize the velocity with unsupervised. You can see they're going much faster, still with a wide diversity of colors, shape, and so on. And here is both unsupervised. So basically here we try to have patterns that are very stable. So that's why most of them don't move. That's what we want. That's what we optimize for, but still they're very diverse. So the dimensionally, we can use dimensionality reduction techniques to project those individuals into a 3-dimensional space, because right now they are embedded in a 6-dimensional space. And we can visualize the population like this with t-SNE, for example. And I really recommend checking out the website so that you can explore this cloud yourself and see even like the behavior of each individual in the population. So finally, I will conclude with results that show some hallmark of open-endedness in this algorithm. Here I show 3 metrics. So we have the entropy of the population, the variance, and the cumulative number of individuals added in the population. And you can see that the entropy and the variance are increasing constantly during the run. So it shows a growing information richness within the population, and show that basically we are continuously feeding the population with new phenotypes that are diverse. So yeah, in conclusion, our findings not only showcase the breadth of artificial life within Lenia, but also underscore the relevance of quality diversity methods in illuminating an ecosystem of artificial species and exhibiting a sustained generation of diversity. Yeah, so quality diversity is an effective framework for the automatic discovery of diverse self-organizing patterns in complex systems. And it has the potential to present some hallmarks of open-ended evolution, aligning with its original purpose. And yeah, don't forget to check out the website. There are way more experiments that you can explore for yourself. And yeah, we encourage the community to participate and build on top of this framework to discover even more diverse and novel life forms. So, thank you.